Hi, my name is Avery Savoy, and today I'm going to be discussing the mitochondrial genome of Tapirus pinchaque. For starters, this is the tapir, the genus that includes Tapirus pinchaque, as well as three other extant species of tapir, Tapirus terrestris, Tapirus indicus, and Tapirus bayardi. The tapir is a relatively large animal that usually ranges from 330 pounds to 700 pounds. Every species in this genus is herbivorous and has an elongated proboscis, which it uses to eat hanging foliage. They are also odd-toed ungulates, with hooves and 3 toes on their back feet and 4 toes on their front feet. Tapirus pinchaque is the smallest of all the tapir species, ranging from only 330 to 500 pounds, with the females being usually about 50 pounds heavier than the males. It lives in mostly temperate climates and at altitudes ranging from 1,400 to 4,400 meters. It is also called the woolly tapir because of its thick, coarse fur, or the mountain tapir because it's native to the Andes mountain range of South America, which is located here. The estimated population of this species is only 2,500 surviving individuals worldwide, as it has fallen victim to deforestation that has destroyed much of its habitat, as well as hunting and removal from its native habitat for placement in zoos. Because of this, the International Union for the Conservation of Nature has identified the species as globally endangered. This study produced the first mitochondrial genome assembly and analysis for this species, which can also be used to aid the conservation efforts of the mountain tapir by providing information to estimate demographic history, heterosis, and population genetic differentiation. This is the mitochondrial genome of Tapirus pinchaque, which is 16,750 base pairs in length. It contains 13 protein coding genes, 22 tRNA genes, 2 ribosomal RNA genes, which are rRNS and rRNL, and 1 putative control region, which you can see here, as indicated by the red arrow. Analysis of the 13 protein coding genes observed 11 starting with a traditional start codon ATG, and NAD3 and NAD5 starting with ATA. For the stop codons, 8 of the PCGs ended with TAA, 3 ended with TAG, and 2 ended with AGA. While AGA is not a traditional mitochondrial stop codon, it was also observed in the same PCGs of Equus cabilis, which is the common donkey. There were no incomplete stop codons observed in this genome. Feel free to take a pause here to take a closer look at this information that I have attached, which is the codon usage analysis for the entire genome. There was a strong AT bias present in the mitochondrial genome of Tapirus pinchaque. The nucleotide compositions were 32.3% for A, 29.9% for T, 25.5% for G, and 12.3% for C. This bias is also visible in most frequent codons of this mitochondrial genome, which were, in order of descending frequency, CTA, which codes for leucine, ATA, which codes for methionine, and ATT, which codes for isoleucine. Visualizations were also performed for the 22 tRNA genes, which ranged in length from 59 base pairs for serine 1 to 75 base pairs for leucine 2. The cloverleaf structure was predicted for all of the tRNAs, which is the 3-arm structure you can see in all of the genes here, apart from serine 1. This missing arm, however, was also noted in the tRNA visualizations of serine 1 of the Rhinoceros unicornis, or the Indian rhino, which is another species in the Perissodactylia order. The putative control region has 1,302 base pairs in length, and presented an AT bias consistent with the rest of the mitochondrial genome. It also had 8 microsatellites and 1 tandem repeat. The selection pressures of this protein coding gene were also looked at. This looks at both the rate of synonymous and non-synonymous mutations in each gene, by comparing each gene to that of Tapirus terrestris. The synonymous mutation rate is the Ks variable, and it is the rate of silent mutation in each gene, while the non-synonymous mutation rate is the Ka variable and represents the mutations that result in a single amino acid change. The ratio of the two, Ka over Ks, is the selection pressure, which is used to estimate the selection pressure of a given gene, enhance gene predictions, and determine differences in natural selection. A Ka-Ks ratio under 1 indicates purifying selection, while a ratio larger than 1 indicates diversifying selection. Strong purifying selection, as exhibited by NAD4L and the 3 COX genes, COX1, 2, and 3, shows a decrease in genetic diversity. The high Ka-Ks ratio of ATP8 indicates weaker purifying selection, which is seen in most vertebrate mitochondrial genome analyses of selection pressure. This phylogenetic tree, which is a maximum likelihood analysis, was developed by my advisor, Dr. Antonio Baeza, using the protein coding genes of this mitochondrial genome. This is the first time that Tapirus pinchaque, as you can see here, is included in the Perissodactylia order. This shows the Tapirus pinchaque as a sister to Tapirus terrestris, as well as a sister-clade relationship between Rhinoceratidae and Teporidae. Finally, this confirms the monophyly of Perissodactylia. In conclusion, this was the first complete mitogenome analysis for the mountain tapir, which will provide information for future metapopulation connectivity research, which can be used in the conservation efforts of this endangered species.